I love lysine, I'm not gonna lie. And in today's post, I wanna tell you why. So lysine is one of the amino acids or protein letters, and it's a cool one. So it is the first one that we've talked about that is charged at our body's pH. It's normally positively charged. We call these basic amino acids, but it's not very basic in terms of the cool things that it can do. And so often its charge gets the most attention, but it can also act as its deprotonated form can act as a strong nucleophile. This is going to let it attack things to form covalent bonds. This is going to allow it to like hold on to cofactors in enzyme active sites to get acetylated, methylated, which helps regulate our gene expression. It can be used for like cross-linking proteins to DNA. There's all sorts of really cool stuff about lysine, and so let's dive in.
So day 12 of 20 days of amino acids, and today is all about lysine. So like all amino acids, it has a generic backbone that has an amino group and a carboxyl group, and then a unique side chain. And so all of these amino acids have these unique side chains that have different properties that are going to influence how the protein folds and functions. And so lysine is here. It is one of the what we call basic amino acids. We'll get into why in a minute, but it's typically positively charged. And this charge is going to let it form charge-charge interactions. We often call, so when we have like full charge interactions, we call these like ionic bonds. So they're not strong bonds, like the bonds that are holding together all of these atoms. They're more just like really strong attractions. And so it can do this, it can form these salt bridges with the acidic amino acids, so like aspartate and glutamate. So this can help kind of stabilize regions of a protein. It's a little stronger of an interaction than you would have with just a partial charge based attractions. And it can also form like salt bridges and stuff with like the backbone of DNA, which is negatively charged and that sort of thing. So it has this this positive charge is coming from this epsilon amino group. And so this is the alpha amino group, and this is the one that all of the amino acids have, but then they lose it when they link up to form these, when they link up in peptide bonds, they're going to lose that end amino group because it's being used to form this peptide bond. So they're only, when you link up amino acids, they're only going to have an amino group, a free primary amino group at the end, and then wherever you have a lysine. So what do I mean by primary amino group? So to classify an amine, you see how many carbons and nitrogen is connected to. So if it's connected to 1, it's a primary. If it's connected to 2, it's a secondary. And if it's connected to 3, it's a tertiary. Almost all of the free amino acids have this primary amine, except for proline, which we looked at the other day, and proline had a secondary amine because it was attached to 2 carbons. But lysine, it's going to have an amine at a free primary amine at its end. And we call this an epsilon amino group because it is at the epsilon carbon. So we use Greek numbering. So we have the alpha carbon, which is the one that like the side chain starts at. And it's also the one connected to like the carbonyl and the nitrogen. Then we count down, so we have beta, gamma, delta, epsilon. And so it's attached, this amino group is attached to the epsilon carbon. And so we call this the epsilon amino group. So we'll talk more about what this means later, but basically it has a pKa of around 10.5. So this means that at a pH of 10.5, half of it is going to be deprotonated. When you go 1 pH below or above the pKa, you're going to change that tenfold. So if you're going below the pKa, if you're at a pH below the pKa, then you're going to be more protonated. So you're going to have more of this positively charged form. And so our bodies are typically at like a pH of like 7.4 or so. So we're going to have about 1,000-fold more of the protonated form than the deprotonated form. But the deprotonated form can still have important functions. And so this deprotonated form can be helped to form if you have the lysine like situated in like an enzyme pocket where there's some basic residue that can help like pull off the proton. And we'll talk more about all this. I just want to give you an overview for people who just kind of want the bigger picture details. So then this, when it's deprotonated, you have a nitrogen with a lone pair of electrons. So you can't see it here. But when you have this, then it's going to allow it to act as a strong nucleophile. And this is going to allow it to form covalent crosslinks with things. So covalent bonds, remember, are like the strong bonds. So these are the ones that are like holding these atoms together. These aren't just like attractions. These are ones where you actually like put a line there in your diagrams. So it can form a couple of different types. It can form like isopeptide bonds, which are like the proton, like the bonds holding together the amino acids, but these are holding together like amino acids from the lysine chain. And this is going to be how you like add ubiquitin to tag a protein for degradation through an isopeptide bond. You can also form these things called shift bases, which is where you have like a nitrogen double bonded to a carbon with the carbon on the other side. And this is going to allow it to hold on to enzyme cofactors like PLP that we'll look at. And so this is going to allow it to hold on to those cofactors, so like a vitamin that's going to help the enzyme work. And so this is going to help keep a tight hold on that. And we'll also see that it's going to allow it to be like cross-linked using like formaldehyde to try to see where a protein is binding, all sorts of cool stuff happening with lysine. And this is exploiting its nucleophilicity. And we'll also talk about like the basicity and how it acts, why we call it a base because it's acted as a base and taken a proton. So in this state, it's actually its conjugate acid state, as we'll get into. So it's coded for by AAA and AAG. So it's lysine. Okay, so ready to dive in? Hopefully, or else you're probably just left. Okay, let's go. Let's start with this charge. So atoms, so these atoms are all linked. These atoms all get, so molecules are going to get their charge from an imbalance of electrons and protons. So much more on this in other posts, but basically all of those atoms, so all of those carbons, nitrogens, hydrogens, they're made up of smaller subatomic particles. So there's positively charged protons, and the number of protons is going to define the element. These positively charged protons are kind of glued together with the help of these neutral neutrons, and they're hanging out in this tiny little central nucleus that's really dense. And then you have this big electron cloud where these negatively charged electrons are whizzing around. These are equal and oppositely charged to the protons. So if you have an equal number of protons and electrons, you have a neutral molecule. But the number of electrons can change, and atoms can like share and swap pairs of electrons. So when they share a pair of electrons, that's how we form a bond. So a pair for a single bond, two pairs for a double bond. And they can also like give and take electrons. So you can end up with an imbalance of protons and electrons. So the protons are like fixed number, but the electrons can change. And so therefore you can get different charged molecules. And when we have a charged molecule, like a fully charged molecule, we call it an ion. You can also get molecules where like it's partly charged because basically when they're sharing those pairs of electrons, they're like merging parts of their electron clouds. And the electrons can kind of like hang out more in one part than the other. And so they'll hang out more around like the more electronegative atom. So the one that's like better at using its protons to like pull and rein in the electrons. And so like oxygen's a really good example. And so is nitrogen of like an electronegative atom. Okay. So that'll give you like a partial charge, and then you can have charge attractions, you can have partial charge attractions, you can have partial charge attractions, all sorts of ways that molecules can interact. And these are all just through like attractions. So sometimes we call them like hydrogen bonds or ionic bonds, but really those are just stronger attractions. So we can talk about getting an imbalance of protons and electrons. So it can be kind of confusing when we talk about like protons, because we're just talking about protons being these like positively charged things that all of these atoms have. But so it turns out that hydrogen, it only has a single proton. So it has this like atomic number of 1. And so because it only has a single proton and in like its most common form, it doesn't have any neutrons, basically in its neutral form, it would have one proton and one electron. If it loses that electron, now it's just a proton. So we can use H plus interchangeably with proton, and we're still talking about the same thing. And so that's why we're going to see H plus used. We're going to see H plus used when we talk about like how lysine is going to be charged or uncharged. And this idea of pH. So a pH is going to be an indication of how many protons are around, so like the proton concentration. But it's kind of confusing because it's an inverse log. So basically, the more protons there are, the lower the pH and the more acidic. The fewer protons there are, the higher the pH and the more basic or alkaline. A couple other terms that we use are a base is something that accepts protons, so it'll take a proton, and then an acid is something that donates protons, so it will give a proton. And you can kind of think of the pH as being a kind of overall measure of the proton availability. So I like to think of it as like a proton cookie jar, where the level, the pH is going to depend on how many like acids and bases you have. And then like how like greedy they are. So there are 3 amino acids that are often positively charged, and then 2 that are often negatively charged. And so the 3 positively charged ones, we call these basic because they've acted as a base and they've taken a proton in their like the form we normally find them in. And so the basic is referring to in their neutral form, they act as bases and take a proton. With the acidic amino acids, in their neutral form, so these have this carboxylic acid group in their neutral form that acts as an acid and gives it up. And so now it's what we call its conjugate base. So if you give up so when an acid gives up a proton, it becomes its conjugate base. And when the base takes a proton, it becomes a conjugate acid because they're just like flip sides of the same coin. So if you give it, you can't give it until you get it back. And so you have to take it to get it back. And so you have to act as a base to get it back. So it can get kind of confusing when we talk about the terminology of these being acidic and basic, but we call the positively charged ones basic and the negatively ones charged acidic because we're talking about in their neutral form, they acted as an acid or they acted as a base, but we'll typically find them in their charged form. Although it's less all the time in terms of histidine, but lysine and arginine are always going to be positive. So how do I know this? So we need to look at a value called the pKa. So the pKa is the pH at which half of an acid will be in its protonated form and half will be in its deprotonated form. So if we go above the pKa, if we go above so to a higher pH, we're going to have fewer protons available because of that inverse relationship between proton concentration and pH. If you have fewer protons available, then there's fewer for these to take, and so it's less likely to be protonated. If you go below the pKa, then you have more protons available, so it's more likely to be protonated. And so if you have a pKa of like 10.5 and your pH in your body is like 7.5, then you're about 3 pH units above your pKa. You're about 3 pH units below the pKa. For every pH unit below, you're going 10-fold less. So that's about 10 times 10 times 10. So you have about 1,000-fold more of the positive charge to the neutral form of lysine. With arginine, you have even more of the positively charged. And I know this because it has a higher pKa. And so basically these molecules, each molecule is going to have its own pKa. And so the higher the pKa, the more basic it is. So that means that the pKa is for the conjugate acid. So basically it's saying that in its acid form, so this positively charged form, so that's able to act as an acid, that's able to donate a proton, how likely is it to do so? And so if you have a lower pKa, that means it's a stronger acid. And since correspondingly, it would be a weaker base. if you have a higher pKa, that means it's a weaker acid and a stronger conjugate base. And so when we have a stronger base, we're going to have a higher pKa. And so this is going to have a higher pKa. And so this is almost always going to be positive, and this is almost always going to be positive. Although the pH, although we talk about like the overall pH as being around like 7.4 or so, if you think about like in the pocket of an enzyme or something, there could be amino acids nearby that could be that could help like pull off, that could help act as a base and kind of remove the proton. And so you can get this deprotonated, and this is going to allow it to act as a strong nucleophile. So we talked a lot more about this the other day, but basically a nucleophile is something that has more electrons than it can comfortably handle. And it's looking for something that it can kind of like share electrons with, share that burden of the electrons. So it likes having those electrons because it's going to give it its like full outer shell of valence electrons. But it doesn't like having like, it basically overload bit off more than it can chew. And so now it's looking for something to help share all that negative stuff from the electrons. And so where do you find that help to share your negative stuff is in the nucleus where the protons are. So it's looking for a nucleus of another atom. And so we call it a nucleophile, so a nucleus lover. And those, for what it's looking for is basically it wants to look for something that wants more electrons. And so those things are called electrophiles. And so nucleophiles can attack electrophiles, and this allows you to like form new bonds and that sort of thing. Often through like substitution reactions. So when we there are several like nucleophilic amino acids. So we talked about like serine and threonine and tyrosine. And cysteine, also lysine and histidine. And for these guys, they have to be deprotonated, which is why they often need like an enzyme help to do so. And so, but they have this lone pair of electrons, and these are highly nucleophilic in this state. And this is going to allow them to form like bonds to other things, and to form like post-translationally modified, as well as that, other things. So basically post-translational modification, this is where you're making changes to the amino acids like after they've been added into the chain. And so a quick terminology note, so when we link these up, because we're losing like the amino and the acid parts when we're linking them up, then we often call like the leftovers residues, and so we can refer to these as like residues when they're in the context of like a polypeptide or a protein. So these residues can get modified like after they've already been added to the chain. So for lysine, there's different modifications. It can get methylated, so it has these like CH3 groups added on. It can get acetylated, so it has this group. We'll talk more about these guys and how they regulate your like gene expression through epigenetics. It can also get like hydroxylated, so have an OH group put on, like kind of like we saw with proline. It can get oxidized, so that amine group can get turned into like a carbonyl, and that's going to allow, that happens in collagen, and so it's going to help form like crosslinks in collagen, like hydroxylysine, lysine crosslinks, some pretty cool stuff. So let's look a little closer at this histone modification. So this is kind of like an oversimplification, but basically you have a lot of DNA. In order to get it to like fit inside of your cells, it needs to be wrapped up. And so it's wrapped up around these like proteins called histones. And these histone proteins, so there's like octamers and stuff, and then the DNA kind of wraps around it. And the way it's able to stay wrapped around is because these histones have a lot of lysines. And so when you have a DNA, it's got this negatively charged backbone. The lysine has this positively charged side chain, and this is going to allow this to kind of like help like shield the negative charges of the DNA from each other, so they're not like repelling, and they're okay being wound up like this. And so these lysines are gonna keep things tight. These lysines can get modified, and this modification, it can alter the charge of lysine in some cases when you have like acetylation, and it can also add like bulk, and it can serve as like binding sites, binding marker, like form binding sites for various modifying protein, for various like transcription factors, so things that are going to get the gene expressed. So when you have like your DNA gets transcribed into a messenger RNA copy, which gets translated into protein. And so if you transcribe more, you're going to make more mRNA copies, and that can be used to make more protein. So by making these like epigenetic changes, so we're not changing the DNA itself, we're not changing the genes or anything, we're just adding this extra information to tell the like cells like, okay, you should transcribe this now, or maybe not. And so basically there's these two main forms. So there's methylation and acetylation. I should note that these lysines, so there's actually a lot of lysines in the tails of these histones. And so you can have these modifications happening on different lysines in like different histone proteins. And so you can have all sorts of various modifications. There are some like more like canonical ones, other ones. Some of them are still kind of unclear what they do. Some are more well established. But basically, so methylation, remember methylation is going to maintain the charge. So when you're methylating it, it's still going to maintain like a positively charged. And so it's still going to be able to interact with the DNA. It's bulkier, so that could disrupt things a little bit. So it was originally mainly thought that it was mainly like suppressive, but it turns out it's actually, the effects will vary more. It depends on like where it occurs. So like what lysine residues, what histone proteins, what transcription factors are going to bind it, and that sort of thing. So all sorts of various things, but there are like special domains of like some regulatory proteins that can recognize like methylation and acetylation marks. But basically, so methylation will maintain your charge, but acetylation, it basically like acts as your charge, you can remember. So acetylation acts as your charge, so transcription factors can gain access. So basically, when you have acetylation, you're going to neutralize the lysine group, amino group, and this is going to make it neutral. And if it's neutral, it's not going to have those charge attractions to the DNA. And so this is going to help loosen up the DNA so it can be transcribed. So when you see that when it gets acetylated, it's going to have this like amide bond. So it looks kind of like those peptide bonds we saw. But it has this like acetyl group here. And so this is going to be resonance stabilized. And because it's resonance stabilized, basically this lone pair of electrons that you would think, why doesn't it grab a proton? Lone pair will be shared into this peptide bond through resonance. So basically, although we're drawing the double bond on the oxygen, we can draw like resonance structures. So basically the lone pair of electrons can kind of like donate into here. So the electrons are shared like throughout here. And so because they're being shared, they can't be used to grab a proton. And so you're not going to see that protonated, at least not like past the one proton it already has. So that lone pair is not going to protonate, and so you're not going to have the charge. And since you're not going to have the charge, it's going to weaken the grip on the DNA and allow for access. So what is this acetyl group? So basically this is an acetyl group. It is the simplest acyl group. So this is like an acyl group where you have a carbonyl carbon. So basically a C double bonded to an O bonded to some other carbon containing group. In the case that we're talking about, we're going to have this acetyl group. So it has a methyl group bonded to a carbonyl. And it's going to get transferred to the lysine through this molecule called acetyl-CoA. So this is a really common molecule that'll come up a lot in biochemistry because it's going to act as like a donor for both acetyl groups, as well as it can also serve as like a transfer of other acyl groups. So you'll see it in like lipid breakdown and that sort of thing. So this basically the CoA part is this, it's derived from vitamin B5 or pantothenate. And it's attached to this acetyl part through a thioester linkage. So basically this, you have a thioester is where you have, so an ester would be if you had a double bonded to O and then an O there. When you have an S there, this is a thioester. So you have the sulfur equivalent of an ester. And this is going to be, it's kind of like swappable. So basically you can have this be attacked. And so you can have, we're going to see the lysine can attack here and then basically steal this, leaving you with CoA. And so because this is through this thioester linkage, sometimes we abbreviate this like acetyl S-CoA. So this is the same, this is just the same thing. Like you're not adding an extra S or anything. This is just emphasizing that the linkage is coming through the sulfur. And so then we often call like the spent form CoA SH. So basically it has spent this acetyl group and now you just have this thiol group. So this SH here. And then this can be regenerated. So you can add another acetyl group on it, including from pyruvate. And so we saw this when we were looking at the tricarboxylic acid cycle, aka Krebs cycle or citric acid cycle, that when you take pyruvate, you can generate acetyl-CoA, and that can ensure like the tricarboxylic acid cycle to be used to make like energy or glucose and stuff. So we often think about acetyl-CoA as being like in context of this cycle and in terms of like lipid breakdown, but it can also be used to acetylate proteins. And so lysine's epsilon amino group is going to let it do this. And so basically, remember this nitrogen is going to be a strong nucleophile, and so it can attack things. And so one of the common things that a nucleophile will attack, so remember a nucleophile is going to want to attack an electrophile. So something that is often like partly positively charged or fully positively charged, and it's looking for some electrons to love it. And one place that one electrophilic thing is this carbonyl carbon. So basically the oxygen, since it's so electronegative, it's kind of like pulling away the electrons, leaving this partially positive. And it's like, dude, I want some electrons. And so this nucleophile can attack it. When it attacks, you end up with like this weird tetrahedral intermediate. And then this can collapse and you can get proton transfer with the help of water, leaving you with this acetyl lysine and this CoA. And so we have this like amide bond here. And then our CoA, which can then be like regenerated and reused. And so you can imagine like similar types of linkages happening to allow lysine to link to other things. And so one place that you'll see this is with ubiquitination. So basically the ubiquitin proteasome system is basically you can tag a protein with this like ubiquitin, which is another little protein. You can get like chains of ubiquitin, and this is going to send the protein for degradation in this complex called the proteasome. So there's like, it happens through these like ubiquitin ligases are going to recognize the substrate and then like bring in the ubiquitin and add these ubiquitins and then take it to it'll get sent to bind to the proteasome and then get it all chewed up. But the reason why I'm telling you this is because this ubiquitin, so these little ubiquitin proteins, they're actually going to link up to other ubiquitin proteins to form this chain through lysine. So lysine can link up through like the C terminus of one ubiquitin protein to another ubiquitin protein. And so there's a couple of different places it can do this, but basically it can form these isopeptide bonds. And so because it's like we're making a peptide bond because remember we have like this C terminus and it looks like an N terminus, but it's not, it's the end of a lysine. And so this is called an isopeptide bond. Another type of bond that we can see lysine make is called a shift base, which is basically where you have a nitrogen double bonded to a carbon and then it has like a carbon on either side. And this is another kind of linkage that's kind of swappable. So we talked about how the thioester was kind of like swappable. This shift base is also kind of like swappable. And so this is going to allow lysine to like hold on tightly. So it's covalent, but it's not like permanent. It can be swapped out more easily than say like breaking a peptide bond or that sort of thing. So basically, if you have an enzyme, so in this case, our enzyme is transaminase. So this transaminase enzyme, basically what it's going to do is it's going to help with the breakdown of amino acids to like get rid of their amine groups in a safe manner. So we talked about like the glucose alanine cycle when we're talking about alanine and how you can remove the amino group like in your muscles from amino acids, and then can come through the blood as alanine into the liver, where they can get that can get passed off and get excreted safely as urea. Is pass off of amino acids like from the glutamate to the pyruvate, then from the alanine to the alpha ketoglutarate. Basically, this is happening with this ALT enzyme. So in this case, we're talking about alanine aminotransferase, so ALT, but there are a bunch of other aminotransferases that use this, but there are other aminotransferases that are like using the same sort of mechanism. But here we're talking about ALT, and the cool thing about ALT is that your liver has a lot of it. And so if you have liver problems, then this ALT can kind of like leak out, and you'll have high levels of elevated ALT in your blood, and doctors can use this to kind of see that your liver is having problems. But hopefully that doesn't happen. And it's active site, so like the place where the things are going to happen, so where the swap is all happening and everything's binding, all the cool stuff, the active site, it has this transaminase residue sticking out, or it has this lysine residue sticking out. And then it's going to bind to this cofactor, so like this helper molecule called pyridoxal phosphate. This is another like vitamin-derived cofactor. This PLP is going to form a shift base with this transaminase. So shift bases can form when you have like an amine group and like an aldehyde or ketone. And so here we're going to have the transaminase attached to the PLP through the PLP's aldehyde group. And so an aldehyde is where you have a carbon double bonded to an oxygen and then another hydrogen. So it's like a terminal carbonyl group. So they can form this shift base, and then this is like held in place. Then a free amino acid comes in, and so our goal here is to transfer this, get rid of this amino group, and ultimately transfer it to alpha ketoglutarate to form glutamate. So we're trying to do this step. We're talking about taking alpha ketoglutarate, taking an amine group, sticking it on alpha ketoglutarate to make glutamate. So we have a free amino acid come in. So in the case of the glucose alanine cycle, that was an alanine we were looking at here. It can be any free amino acid. And basically what's going to happen is you're going to swap it out. And so the transaminase, so now this amines, so this is the shift base is also called an imine. And this basically can get swapped out. So you can swap out this free, this shift base for this shift base. So instead of forming a shift base to the transaminase, now we're forming a shift base to this amino acid. So it's no longer like covalently held in the enzyme site, but there's still a bunch of other attractions and stuff that are holding it there for things to keep going. What's going to happen now is that this bond is subject to hydrolysis. So basically water is the enzyme is going to help water cut it free. And so this is going to leave you with an alpha keto acid. And then the amine group transferred to this cofactor. So this is now going to be PMP, pyridoxamine phosphate. So if we follow the amine group, we are taking it from the amino acid, we are using it to link up to this PLP. Now we cut off the rest of the amino acid, leaving behind the amine group. And so now it's on this cofactor. And so now that we've like gotten rid of the amino group from the amino acid, so now it's an alpha keto acid. So it has a ketone, so a carbon double bonded to an oxygen with carbons on either side. And then this, which can be used in various forms depending on like which amino acid it was, it can enter various metabolic pathways. But so now you have this PMP. So now we have the amino group, we've transferred it, but it's still stuck on this. And so we want to get rid of it. And so we can get rid of it by moving it to alpha ketoglutarate. And when we do that, we make glutamate. And then this PLP is regenerated. You have this PLP regenerated because you removed that amino group. And so now we can use it again in this reaction. So we've talked about like the like in your body uses it this way, but we can also exploit lysine's nucleophilicity in the lab to allow us to cross-link things to it. So basically when we talk about cross-linking, we're talking about like forming covalent bonds from one molecule to like another, to a different part of the same molecule, or that sort of thing. And so we can use formaldehyde to form like protein-protein and protein nucleic acid, so like DNA or RNA cross-links. And this is going to happen through a shift base as well. So in this case, instead of attacking that's like PMP, we are going to be attacking this the aldehyde of the PMP. We're going to be attacking formaldehyde. And so it's the same sort of thing. Where you're getting this shift base, and then this is going to allow you to then have a second thing attack the shift base, and then you end up with a cross-link. So here we're not like swapping out the shift bases, but we're using a shift base intermediate in order to like attack and then get this second linked, get this cross-linked product. You can also use it to get information about binding to like DNA or RNA. So like ChIP-seq, chromatin, cross-linking chromatin amino precipitation, or cross-linking histone amino precipitation. I can't remember exactly what it stands for right now, but there's a bunch of different varieties of it. But the basic idea is that you can cross-link a protein to like DNA and then or RNA, and then cut around it and then sequence the pieces. What's really great about this is when you're trying to do like DNA binding, you can use it to like figure out like where histones are bound. And this can give you information about like the relative accessibility and various things of regions of DNA, and like what sort of modified histones there might be in that sort of thing. But why this is really useful is because your histones, remember, they have a lot of those lysines, and the lysines are what's going to be reacting with like the formaldehyde and stuff. So you can form crosslinks where you want them. When you want you can form crosslinks more easily with those proteins. In addition to formaldehyde, there are other cross-linkers that we can use, and some of these are like mass spectrometry compatible. So the cross-linkers that we typically use are like homo or hetero bifunctional. So basically they have an attacking group on either side. And this is going to allow you to cross-link protein to protein. So within the same protein or within different proteins. So more on this in a post before, but here's a quick little recap-y type thing. And then I'll show you some of the other things we can do with like conjugation. It's kind of like if you're doing like protein-protein, you can learn about like where various parts of different molecules are interacting, or various parts of the same molecule. So basically you do this cross-linking, and then you can use mass spectrometry to kind of like cut the pieces apart, and then determine the mass of those things using mass spectrometry. So basically find the size of these fragments and then their sequence, use that to get their sequence, and then figure out like where the identity of them, or where the cross-links happened. You can also use it to kind of just stabilize a complex if you want to use like structural biology, like cryo-EM or crystallography, in order to try to like stabilize a complex in order to like get structural information about it. In addition to just like using something kind of like generic to cross-link lysine to what's already around it, we can introduce something around it that's attached to something that we know lysine can attack and get stuck to. So a common way, this allows us to like make conjugations. So basically make covalent bonds between a lysine and something else. So this could be something else. This something else could be like a fluorescent label. It could be a bead so that you can like for chromatography or even precipitation something. Or I guess it wouldn't be, I mean, it can be like a bead for some sort of like precipitation or something. Some you want to stick it to a chip, various things that you can do. And so these rely on this like amine reactive crosslinker chemistry, basically. And a common type is like this NHS esters or N-hydroxysuccinimide esters. So I talked about these a little more when I was talking about crosslinking before. But it's this group here, this NHS ester. And so basically you have this ester, it's stuck to something. You have your primary amine on your protein. So this could be on the N-terminus or it could be on a lysine. And then it's going to form an amide bond. And this NHS group is going to go bye-bye. So this nitrogen can attack this carbonyl carbon like we've seen before, and then this is going to leave as your leaving group. And you're going to have this amide bond. And so this is going to allow you to do some cool stuff. So there's different variants of it, like sulfur esters. There's ones that are mass spec compatible. So you can cross-link things on your protein and then use mass spec to figure out where they were binding. You can conjugate your protein to like antibody HRP, or I guess they're often used to like conjugate antibodies to things. And so you can conjugate antibodies to like a fluorophore or something, or conjugate your antibody to some sort of resin or bead if you want to do some sort of like immunoprecipitation. And so basically whenever you want to get your protein stuck to something, then you can take advantage of lysine's ability to form these sort of conjugations to react with these NHS esters. And so we saw it reacting with like the thioester when we were talking about acetyl-CoA, and here we're talking about an ester. So that's where you have an oxygen instead of that sulfur there. But it's the same idea. And so there's also other various versions of things that you can do to react to a lysine and to react to the N-terminus. And so, yeah, this is just Thermo Fisher has a bunch of information about like cross-linking and that sort of thing. Yeah, so this is an ester. This is an ester, so it has this carbonyl oxygen. So then we've looked a lot at amides, which have a nitrogen, and then the thioester is where this would be a sulfur. And so we see the thioester linkage when we're looking at acetyl-CoA. And when we're looking at those NHS esters, we were looking at an ester. But either way, you have this carbonyl carbon where the oxygen is pulling electrons away from the carbon. And so this is making this carbon electrophilic, and it can serve as an attack point for the nucleophile, like the amine, the deprotonated amine of a lysine or the N-terminus of a protein.